about the bile acids in the feed of shrimp and carnivorous fish. And bile acids are the important components of the bile. And bile acids have many critical functions, such as it can enhance the fat emulsification and enhance the lipid catabolism, and also enhance the digestion of the lipid-soluble vitamins. And it can also produce the bilirubin. In this picture, we can see that bile acids can produce from the cholesterol and from two pathways. One is the classical pathway, and the other is the alternative pathway. And the bile acids mainly synthesized in the liver, and it can transport to other tissues. And with the help of the microbiota, it can produce to the secondary bile acids, and the bile acids in the intestine can also being reabsorbed to the liver, and here we call it the liver intestine circulation. And here are the bile acids chemical structure. We can see that for the different kinds of bile acids, the difference is mainly the three side chain group. The different side chain group and being the different kinds of bile acids. And the bile acids mainly can be two types, primary bile acids and the secondary bile acids. And here is the many pathway about the bile acids metabolism, and we can see that the bile acids can enhance the fatty acids fat oxidation, and it can also inhibition of the glucogenesis. And it also have important role in the anti-inflammation. And the center of the bile acids metabolism is the FXR1 bile acids receptor. Here is a review published in last year. And in this review, we can see that the bile acids application in the fish diet, and it can regulate the inert metabolism and improve the health condition of the fish. And in this figure, we can see that there are many kinds of fish, such as the grouper, the largemouth bass, and the tilapia. There are many kinds of fish have applicated the bile acids in their diet. And here is the suitable level of the bile acids, and in some research, the bile acids are supplemented in the normal diet, and in some research, they are supplemented in the high lipid diet, and some of them supplemented in the high starch diet. And also, there are many research about the bile acids in the vegetable protein-based diet. And here is a review about the research until now. And we can see that most of the research about the bile acids is mainly in the carnivorous fish. And how to use the bile acids? I have concluded that there are several application scenarios. And one is the common scenarios is it is additive with a wide range of functions, such as enhance the improve the growth and enhance the immune response. And there are also 5 typical scenarios. The first typical application scenarios is the application in the high-fat diet. And here is an article we published 3 years ago. And this article is a highly cited paper and is also a hot paper. And this study was conducted in the largemouth bass, a very popular fish in China. And we can see the growth performance with the addition of the bile acids, the growth performance was improved. And we also detected many immune response related genes. We can see the gene expression also enhanced by the supplementation of the bile acids. And here is the lipid in the liver, and we use the oil reader stain to show the lipid contents in the liver of the largemouth bass. And here is the high-starch group. We can see there are lots of lipids accumulated in the liver of the fish, and after we supplemented the bile acids in the diet, the lipid contents reduced. And here is another research we have done 2 years ago. And here in this study, the bile acids was applied in the diet of group. And in this study, we also set the control diet and the high-fat diet, and then we supplemented the bile acids in the high-fat diet. And here is the supplementation level of the bile acids. And we can see that for the growth performance, the final body weight, the weight gain, and the specific growth rate, with the addition of the bile acids, the growth of the group increased, and the suitable supplementation level is in this group is 900 milligram. And in this study, we also detected the lipid contents in the liver of group is similar as the last experiment in largemouth bass. We can see that with the addition of the bile acids in the high-fat diet, the lipid accumulation was reduced. And we also detected the microbiota, and we can see that the bile acids also affect the diversity of the microbiota. And here is the bile acids content in the liver, in the cerebrum, and in the intestine. And we can see that after in the high-fat diet, we can see that the bile acids decreased, and with the supplementation of the bile acids, the bile acids in the liver, in the cerebrum, and in the intestine all increased. So the supplementation of the bile acids increased the bile acids pool in the group. And we also detected the bile acids transport related gene. We can see that the bile acid transport related gene expression was decreased in the high fat group. And after we supplemented the bile acids in the high fat diet, their gene expression was increased. So dietary supplementation of the bile acids enhanced the bile acids transport. And here is the final review of this study. We concluded that the bile acids, supplementation of the bile acids increased the bile acids related microbiota and increased the content of the primary bile acids and enhanced the bile acid transport and reabsorption. And it's mainly through FXR and TGR5, the two bile acids receptors. And then enhance the lipolysis and reduce the lipogenesis. Finally, reduce the lipid accumulation in the group. And other scenarios of application of bile acids is in the high starch diet. And here is also a study in the large mouse bus. And we can see that the supplementation of the bile acids also improved the lipid and glucose metabolism. Since it's similar to the fat diet, so we move to the next scenarios. The next typical application scenarios is in the low fish meal diet, and it's very important. This is a review published 4 years ago, and in this review, the author concluded that with the replacement of fish meal, the bile acids will be reduced and induce some mitigating effects. And these mitigating effects could be reduced by the dietary bile acids supplementation. And why did the bile acid would reduce in the low fish meal diet? We think it's mainly due to 3 reasons. The first is the bile acids contents reduced because bile acids is higher in the fish meal and animal protein, but it's very low or zero in the plant protein. So if you replace the fish meal, the bile acids content will be reduced. And then the second reason may be the increased bile acids excretion and or the decreased reabsorption. This mainly due to the disturber of the anti-nutrition factors. And the third reason is the bile acids synthesis may be reduced in the low fish meal diet. And here is an example in the Atlantic salmon. And here we can see for the Atlantic salmon, if it feed with the high plant diet, and here is the feeding time, we can see that the here is the bile acids in the plasma, here is in the liver, and here is in the intestine. We can see that the bile acids in the Atlantic salmon will be decreased after intake of the heplant diet. But for this, there are also many exciting problems, such as for different kinds of the plant protein. Until now we don't know they have the different kinds of anti-nutritional factors. So about the effects of different anti-nutritional factors on the bile acids is also unknown. And it's also have many differences in the different aquatic animals. So it's the exerting problems about the low fish meal diet. And until now, there are only a few studies about the anti-nutritional factors on the bile acids metabolism. And here is an example. They used one common anti-nutritional factors and to explore the addition out of the anti-nutritional factors and the addition of the bile acids on the bile acids content in the fish, and finally find the bile acids balance. They found that when anti-nutritional factors were supplemented in the diet, the bile acids will be reduced, and it could increase after you supplemented the bile acids in the diet. And here is other applications scenarios of the bile acids is for the detoxification. Here is an article published article in shrimp, and we can see that here they used one common toxin, and after the addition of the bile acids, we can see that the toxin residue will be reduced after you supplemented the bile acids in the diet. And we also found that the bile acid could reduce the inflammation caused by the toxins. And the final application scenarios of the bile acids maybe enhance the fat-soluble nutrients. We know that in the diet there are many kinds of fat-soluble nutrients, such as the sterols, the fat-soluble vitamins, the hilfas, and the astaxanthin. Bile acids can enhance the digestion and the utilization of the fat-soluble nutrients. And I think it's a very important point to reduce the feed cost, because some of the fat-soluble nutrients, they are very expensive. For example, the hilfas and the astaxanthins, they are very expensive. So bile acids can enhance their utilization and would be beneficial to reduce the cost of the feed. And here is an example in this fish, and in this experiment, they cited 3 levels of acetylsins and 2 levels of bile acids. None for this are not supplemented with bile acids, and 3 groups they supplemented with some bile acids. And here is the weight gain of the fish. We can see that when you supplemented the bile acids in the diet, no matter the level of the acetylsins, bile acids would increase the weight gain of the fish. And here is the acetylsin content in the fish, and we can see that it's similar as the weight gain. The addition of the bile acids would increase the acetylsin contents in the back of the fish. So it's very important, and it's very interesting experiment. And another part of the research is mainly about the bile acids metabolism in the crustaceans, and it's for example, the shrimp, it's a very big species in China and in the world. Here are the main shrimp species found in China. And these 4 species are found in the seawater. And this 3 shrimp is found in the freshwater. And in China, the most important shrimp is the little Pinus phenomenon. And in China, the production is close to 2 million tons. And for the second important shrimp in China is the Pinus monodon. The Pinus monodon until now it's about 100,000 tons in China. And as I mentioned before, the bile acids can't be synthesized in the shrimp, but in these years, there are many articles. They find that the critical role of the bile acids in the diet of shrimp. They find the bile acids can improve the growth performance and the lipid metabolism and the immune response and also the intestine health of the shrimp. And in the past 5 years, I have done many research in the shrimp about the bile acids. Here is my first research about the bile acids in the shrimp. In this experiment, we set 3 diets. One is the high fish meal diet, and the other is the low fish meal diet. And the third one is added the bile acids. Here we use the CDC. We added the 0.1% bile acid in the low fish meal diet. And we can see that here is the growth performance. Here is the growth performance. We can see that the growth performance of the shrimp was improved after we added the bile acids in the low fish meal diet. We can see from the final body weight and the weight gain, and also the feed efficiency was increased after the dietary supplementation of the bile acids. And also for the lipid metabolism in the liver, we can see it's also improved by the supplementation of the bile acids. And we also detected the histology of the intestine and the liver. And especially in the intestine, we can see that here is the histology in the low fish meal group. We can see that the intestinal fold heat was lower in low fish meal group compared with the high fish meal group. And after we supplemented the bile acids, the fold heat was increased. And also for the ER swollen, we can see in the low fish meal diet, we find the ER swollen here. And after we supplemented the CTC, this situation was elevated. And we also detected the microbiota of the shrimp. We can find that the supplementation of the bile acids in the low fish meal diet increased the diversity and the richness of the intestinal microbiota of the shrimp, and also reduced the harmful bacteria abundance. And for the last experiment is in the Pineers model then. And we also conducted the similar experiment in the little Pineers Vietnamese. And in this experiment, we used 4 diets, also a high fish meal diet or low fish meal diet, and 2 levels of bile acid. And in this experiment, we can see that it's very clear. Here is the intestine, and we can see that the intestine health would be improved after we supplemented the bile acids in the diet. It's very clear here. And we also detected the gene expression related to the apoptosis. And we can see that the dietary supplementation of bile acids in the low fish meal diet also improved the immune response of the white shrimp. And here is another experiment in the white shrimp. And in this experiment, we used the bile acids provided by luncheons. And in previous study, we used the CDC, but in this study, we used the commercial bile acids. And it is a similar experiment, and we also have the high fish meal data, low fish meal data, and 4 supplementation level of bile acids. And for the growth performance, we can see that with the addition of bile acids, the growth, the final body weight, and the weight gain, we can see they increased after we supplemented the bile acids in the low fish meal diet. And one of the main reasons may be the bile acids could enhance the molecule-related gene expression. We have detected lots of molecule-related gene expression. We found that bile acids could enhance their expression. So this may be one of the main reasons for the growth-promoting effects of the bile acids. And here is an article we published in last year. And in this experiment, we mainly detected the gene expression related to the sterol metabolism and the bile acids circulation related gene expression. And we also found the bile acids could promote the sterol metabolism. And above the study, the research we have done in the last 5 years, and now in the future research we are interested in some directions such as the single cell transcriptomics, and we want to find the cell population and the cell communication changes induced by the bile acids. And we also want to explore the association between the intestinal microbiota and the bile acids metabolism. And we think the intestinal microbiota may be very important to the bile acids metabolism. And we also want to evaluate the different function of the commercial bile acids and the pure bile acids, such as the CDC and the UDC and the different kinds of bile acids. We want to explore the different function of the different kinds of bile acids. So here is the main content of my report, and thank you for your attention.
Thank you so much, Dr. Xie. Dr. Xie introduced the applications of bile acids